Hello, and a very warm welcome to today's Nucleic Acid Insights webinar, titled Engineering Lipid Nanoparticles for Lung Cancer Imaging. I'm Roisin McGuigan, an editor at BioInsights, and joining me today I have Ben Nappa and Blaine McCarthy, who will discuss the use of engineered LNPs to deliver DNA vectors for cancer imaging. After the presentations today, we'll have a live Q&A session. So we invite our audience to pose their questions to our speakers using the Ask a Question box at the bottom of your screen, and we'll try to get to it during the session. I'd also like to draw your attention to the resources on the right-hand side of your screen, where you can find more information on the topics covered today. Next up, I'd like to introduce our presenters. Ben is Product Manager, LNP Solutions at Unchained Labs, focusing on automated, high-throughput lipid nanoparticle encapsulation for drug delivery, vaccines, and gene therapy. Blaine leads Earli's delivery team, leveraging her expertise in polymer and lipid nanoparticle formulations to advance early cancer detection technologies. So without further ado, I'll now hand over to Ben to kick us off with his presentation.
Thank you for the introduction, and thank you all for taking some time to join us today for this webinar, which is going to give you all some insight into how the team at Earli have been using engineered lipid nanoparticles for lung cancer imaging. Natural disasters pose a significant threat to human life. They can come in many forms, large or small. And for a range of different large-scale natural disasters, be it tsunamis, earthquakes, wildfires, or tornadoes, early detection and response is one of the most effective methods of reducing fatalities. The sooner the alert is raised and action can be taken, the more lives are likely to be saved. Now, cancer could be considered as a type of natural disaster on an entirely different scale, both in size and scope. However, the importance of detecting and treating cancer early have just as critical an impact on survival rate. To give you an idea with some statistics, in 2023, there were roughly 95,000 reported fatalities from natural disasters worldwide, and that's one of the highest numbers since 2010. However, according to the World Cancer Research Fund, 2022 saw an estimated 1.8 million deaths from lung cancer alone. The process for alerting is twofold. Step one is diagnostic to identify the cancer, in particular its location and its type. And the second step is therapeutic, treat the cancer before aggressive symptoms develop, and whilst the chances of survival remain high. LNPs offer a hugely promising way to deliver nucleic acid cargo to specific cell or tissue types by engineering their properties, enabling the cargo to be safely delivered in the relevant cell type and released for translation or expression within the cell. Cancerous cells present a particular challenge due to their close relation to healthy or benign cells, complicating the process of generating a positive detection signal from the cancer cell without causing false positives from surrounding healthy cells. The tunability of LNPs can be both a blessing and a curse. The extensive tunability of LNP performance does stem from the complexity of their structure, the range of different lipid types and functionalities that can be incorporated into that structure. And this can make identifying the most promising formulations a little bit like finding a needle in a haystack. So there needs to be a way to screen lots of different formulations with a high degree of accuracy at low volumes whilst maintaining a consistent method of making much larger volumes of material without significant changes to the production process. This is where Sunscreen comes in. So with Sunscreen, you can load up to 96 individual unique formulations, be it different lipid mixes or different cargo types, into 96 well plates on the system's fluid handlers. And these get run through the system at a rate of around 3 to 4 minutes per experiment, taking a pair of samples from those two input well plates, running them through one of our microfluidic mixing chips called Sunnies, and collecting them into a collection well plate with the option of having inline dilution as well. And the volume of the experiments can be as low as 400 microliters for a typical 3-to-1 ratio. And the mixer types, the Sunnies I mentioned, come in a range of different types and sizes, allowing for different sunnies to be used for different applications or different processes. The system has built-in automated cleaning, and this helps to make sure that when you're screening, if you do have any poor formulations, the system immediately will recover for the next good formulation to give consistent results. And you can see some of that from the data on this graph, where we have 90 repeats of identical formulations. And when we have thrown in a bad formulation, like the bars in red, where we've left out the pegylated lipid on purpose, you can see that the recovery from a poor formulation is immediate when we run the next good formulation. The reproducibility of those 90 repeats is less than 1% CV across all of the samples, which means when you do see variations in your results when you're screening, you know that that's down to the formulation changes that you've been intentionally making and not an unintentional variation in the process that the instrument's running. So Sunscreen helps you identify those key formulations, the ones that show the absolute most promise, to identify for further development. And you can do that with Sunshine. So Sunshine allows you to take those ideal formulations and now optimize the process. So Sunshine will allow you to take a pair of reagents injected at a larger volume into the system, still into sample loops in the same way that Sunscreen runs, and will allow you to then automate a series of experiments with those pair of reagents, varying different process parameters such as the total flow rate or the flow rate ratio. So you can see here some data showing a screen across 4 different total flow rates of the system, repeated in triplicates on the instrument, showing a high degree of reproducibility between those repeats. The system is also able to run in a continuous flow mode, which means we can go anywhere from a 1 milliliter minimum experimental volume in the protocol or automated mode, all the way up to a continuous production of up to liters of particles if you wish. The same reusable microfluidics with the Sunnies are usable on both the Sunscreen and the Sunshine system, as well as our GMP manufacturing system, Sunbather. And Sunshine is a perfect system on which to test out those different mixer types and further optimize the formulations you've identified in order to improve them ahead of manufacturing in continuous flow. Because the pumps and the mixers are the same across these systems, the transfer of the protocols you were developing on Sunscreen moves to Sunshine with absolute ease. So without further ado, I'm going to hand over to Blaine, who will explain how Earli have been putting these systems to work over the past couple of years and share some of their fantastic research.
Thanks, Ben, for the great introduction. So I'm excited today to tell you guys a little bit about our company Earli and how we're using the Unchained Labs Sunshine and Sunscreen instruments in order to engineer novel compositions of lipid nanoparticles. So our mission at Earli is to detect and treat cancer at earlier stages. And this is really born out of kind of frustration that, it's been challenging to try to improve the cancer survival rates. And this is partly due to our inability to detect cancer at early stages, such as stage 1. And so you can see that across different types of cancer here, that you can greatly improve patient outcomes just by catching the disease at earlier stages. And so this has really become the mission of our company. And so if we ask this question, how do we detect cancer at earlier stages? We kind of think about this problem as thinking about, instead of finding a needle in a haystack, it's really like finding a needle in a dark ocean. So if you think about the human body, it has all of these cells present. And what we're really looking for is some signature of tumor cells, which might be a very small population of the cells in your body that can shed some sort of proteins or DNA fragments that we can then detect. And so that's the needle. And so if you think about a vast and dark ocean, it's going to be really challenging to find that needle. And so instead, at Earli, what we're trying to do is really flip this question on its head and instead say, okay, if we're searching for this needle, is there a way that instead we can turn that tumor into something that actually guides us towards it? And so we liken this to thinking about turning on a light bulb in a dark ocean, where now we can actually follow this light source and navigate to identify where the tumor is and such that we can actually treat it. And so the question that we've asked ourselves is really, can we develop a technology where we can make cancer reveal itself to us by making those tumor cells actually produce a synthetic protein that we can then measure or capitalize on to actually treat the cancer. And so the way that our platform works is really with four key components to it. The first is that we are using the delivery of a nucleic acid, so a DNA construct, into the tumor cells in order for this technology to work. And some of the key elements of this DNA are first a promoter sequence. And we think about this as essentially an if clause. So our team at Earli puts a lot of work into leveraging bioinformatics data and empirical results in order to identify promoter sequences that are specific for cancer, meaning that proteins that are upregulated in cancer will bind our DNA construct to drive expression. And so if we're in a cancer cell, we can then distinguish and move to the next step. And so the next step there is for that promoter to then drive the expression of really any protein that we want. So you can imagine that for a diagnostic application, we could say express a protein that can be surface expressed to bind a PET-CT tracer. And then you can localize that radio tracer and image it with PET-CT imaging to actually identify where your cancer is. Alternatively, you could also imagine a therapeutic application where now we're expressing something like a cytokine that could recruit the immune system locally to the site of that tumor to actually treat the cancer. Now, of course, for all of this to work, we need to get that DNA construct into the tumor cells in the first place. And that's really where our team, the delivery team at Earli, comes in. So what we've been focused on is using a number of different delivery modalities, but most importantly, lipid nanoparticles in order to deliver Earli's DNA construct. So I know that's kind of a lot of information. So I'm going to try to explain this and walk you through it a little bit with a video. So what I mentioned before is that, we're really encapsulating Earli's DNA into these lipid nanoparticles in order to deliver it to tumor cells. And so the Earli DNA is shown here in purple and encapsulated in this kind of bright green lipid nanoparticle. And so the first key step is for us to actually deliver that DNA into the tumor cells. And once we get that DNA into the nucleus of tumor cells, this is what we're showing as the next step here. So the Earli DNA strand, again, is shown in purple, and that synthetic promoter sequence that's specific for cancer is highlighted in green. And so in the nucleus of a cancer cell, the proteins that are upregulated in that cell due to the genetic dysregulation that makes it a cancer cell in the first place can now bind to that promoter sequence and drive expression. And so if we contrast this scenario now to what would happen in a normal healthy cell, what you'll see is that those proteins are not present or they're not nearly as abundant. And so this allows us to be specific in terms of producing protein only in cancer cells and not in healthy cells. And so again, you can imagine that that protein produced is something that could recruit the immune system to actually kill those cancer cells. Or alternatively, it could be used in a detection application by actually binding a radio tracer that can then be imaged using PET-CT to localize where the cancer is. And so these are the applications of the Earli technology that we're really excited about using to both detect and treat cancer at earlier stages. So if we think about now this problem from the delivery perspective, to use lipid nanoparticles to deliver this DNA construct, there's really two key challenges that we face. The first is that, as many of you may know, lipid nanoparticles primarily distribute to the liver. And so it's estimated that about 70% of an intravenously administered lipid nanoparticle dose goes to the liver. And instead, what we need to do is engineer lipid nanoparticle compositions that will direct that to the tumor such that we get sufficient tumor exposure. The second key challenge here is that we are delivering DNA instead of things like mRNA or siRNA, which need to make it into the cytoplasm. We need to make sure that that DNA actually makes it into the nucleus of the cancer cell, because that's really the locus of the genetic dysregulation and where that synthetic promoter sequence can actually do its job. So as you can imagine, to face these two challenges and overcome them, what our team needs to do is we need to engineer compositions and use lipids that are very different from the types of more traditionally known lipid nanoparticles, such as FDA approved Onpattro, or of course, the more recent COVID vaccines. And so to do this, a key part of our workflow is using the Sunscreen instrument from Unchained Labs. And this allows us to take a broad composition space and rapidly screen through it. So a couple of the things about this instrument that have been particularly helpful for us. The first is that moving from a previous instrument that was completely manual, we found that for one particular researcher, they could usually make about 10 to 15 new compositions of lipid nanoparticles in a day and then carry those forward for testing. Now, using the Sunscreen instrument, we're able to set up a high throughput well plate system and walk away from the instrument while it's running, go do other things, and come back and have between 30 to 80 formulations prepared. So we routinely are now able to up our scale from, again, 10 to 15 formulations per day up to 30 or 80 per run. And the other thing that's beneficial about this is that it allows you to run these formulations at a much lower scale. So it's been about a 3-fold reduction in the amount of material that we need to use for these formulations. So those are two of the things that have made the Sunscreen really helpful for our application. We then take those particles forward and we do biophysical characterization. Now, for the screening approach that we're taking, the thing that we're really focused on here is getting particles that have high encapsulation efficiency. And at this stage of the process, the size and the PDI are not quite as important for us. So then we down select and make sure that we bring forward all the formulations that have high encapsulation efficiency into cell experiments. The nature of the cell experiment, of course, just depends on the research question that we have at hand. But oftentimes we're looking at things like uptake into cells, nuclear delivery, and expression. So I want to give you guys a sense of, what does the data look like? What are some representative examples of what lipid nanoparticles look like when they come off the Sunscreen? So as I mentioned before, we're working with compositions that are very different from Onpattro style or COVID vaccine style particles. So I've just named those compositions here as A, B, and C. A, B, and C are very different from each other as well. And what I'm demonstrating is that across multiple runs, shown in blue and orange for each one of these compositions, we get really good agreement in terms of the size, the PDI, and the zeta potential for these particles. In general, our encapsulation efficiencies tend to be very high, except for, of course, if we're going with very strange compositions, in which case it's more related to the lipids that we've chosen rather than the Sunscreen instrument. So then we can carry these forward into cell-based experiments, and I'll show you just 2 examples of how we do this. So first here, I'm just showing a relatively simple firefly luciferase assay where we've taken the lipid nanoparticles that we made on the Sunscreen, used them to treat cells, and now we're looking for reporter protein expression. And I want to just highlight here that what we did in this particular experiment, we took 3 new ionizable lipids. We put them into very different types of compositions and paired them with different types of helper lipids and sterols. And what we were able to do is rapidly identify some formulations that had high expression in the cell line, again, really with just one set of experiments run on the Sunscreen. Another application that we routinely use the Sunscreen for is to do more complex experiments where we take our DNA and we label it with a commercially available dye. And then we formulate that into our particle using the Sunscreen. And by labeling the DNA, what this allows us to do is to use flow cytometry readouts to actually track now the uptake of our particle into cells, and then the translocation of that DNA into the nucleus as well. And so what I'm showing here on the right-hand side is a plot of a readout from one of these cell-based experiments, where on the y-axis you've got expression, and on the x-axis you've got uptake into cells. And so again, while I can't reveal the specifics of the compositions that we're working with here, I think you can appreciate that functionally we're able to prepare particles that have very different behavior in terms of their ability to get taken up into cells and to express reporter protein. Again, I want to highlight that an advantage of using the Sunscreen is that we're able to use much less material for this. It's particularly nice for these types of experiments where we're using dye-labeled DNA, because we have to make that dye-labeled DNA in the first place. And so it conserves on that material which saves us additional time in the long run. So the piece of this that has also been really nice is that we purchased the sunscreen at the same time that we purchased the sunshine instrument, or that larger scale instrument. And so this has allowed us to move from now these screening experiments that are primarily focused on biophysical characterization and in vitro experiments into actual animal experiments. And so what this typically looks like is moving from that 70 microgram scale that we typically use on the sunscreen. Now we bump that up to about 0.5 to 1.2 milligram scale on the sunshine. And a couple of things that are really nice about this. The first is that we're able to use the exact same microfluidic mixing chip. And so that leads to a lot more translatable results in terms of that particle formation step. And it reduces the need for us to actually go through kind of troubleshooting of the total flow rate and the flow rate ratio by having to move between different mixing geometries on different styles of instrument. The other thing that has been beneficial to us is the ability to put this instrument into continuous mode if you need to scale up for a very large animal experiment. And so we have scaled up to 30 milligrams in-house in order to use that material for other types of experiments. And so from here, we then take those formulations, and for the sunshine workflow, we do a suite of different biophysical characterization each time. So now we're not just focused on encapsulation efficiency, but we are focused on some of these other parameters like size, PDI, and some of the other particle characteristics. So we use those selection criteria now to make sure that the formulation's in good spec for moving forward into animal experiments. So another thing I want to highlight here is that moving from the sunscreen, where we're really in this kind of screening mode, to the sunshine, now it's pretty easy on the sunshine to also change parameters like the total flow rate or the flow rate ratio in order to really hone in on, what's a good PDI and what's a good size for this particle at scale. So here I'm just showing some of the representative data, again, from moving from the sunscreen to the sunshine. So I brought back those three very different compositions of particle, A, B, and C, so shown here. And now what I'm showing you is a comparison of the data from generating these particles using the sunscreen versus generating these particles using the sunshine. And what you can see is that the biophysical characteristics are in really good alignment between the two instruments, including the size, the encapsulation efficiency, and the zeta potential. So overall, this is just to give you guys kind of a snapshot of how we've been using the sunscreen and the sunshine instruments for our application. So the key things that I want to highlight here, the first is just demonstrating that, for our work, we do need to work with compositions that are very different from Onpattro style formulations or those used in the COVID vaccine. And this instrument, for the sunscreen especially, has worked really well to allow us to screen this broad composition space and to work with these very non-traditional lipid nanoparticles. The second thing is that the sunscreen has allowed us to do this at about a threefold lower scale than what we were previously doing, and with a much higher throughput in terms of the number of formulations that we can test at a time. And so this helps us to both conserve material, which, as I mentioned before, for those dye-labeled DNA experiments is important for both the DNA cargo itself as well as for the lipids that we use. And then in terms of exploring this space and to really overcome the two challenges that I outlined in the beginning of distributing to the tumor and escaping the liver, as well as making that DNA prepared so that it can escape the endosome and then also make it to the nucleus, that we have to work with these very different styles of composition. And so the third thing too is that because we have both the sunscreen and the sunshine instrument, that we're able to relatively easily transition from the sunscreen to a larger scale on the sunshine. And that's really nice for us because one, it means that we don't have to do as much troubleshooting in terms of process parameters when we go to scale up for mouse experiments. And the second reason is that because we have both instruments, now we can also do more experiments in parallel, in that someone can be using the sunscreen and screening for cell-based experiments while another person is preparing formulations for mouse experiments. So with that, Ben and I are happy to take any questions that you guys have.
Well, thank you to Blaine and Ben for some great presentations. We're now going to start our Q&A, so I'd invite you to join me on camera. And as a reminder to our audience, do keep your questions coming in. If you want me to direct a question to a specific speaker, you can include this information when you send your question. So first up here, Ben, I have a question for you. At what stage do you switch from using sunscreen to using sunshine?
So that's kind of dependent on a number of different things, but the fundamental difference really between those two instruments is the volume at which you're producing samples. So obviously sunscreen being tailored for the well plate based low volume samples, the maximum volume that can collect is about 2 milliliters of sample per well. And obviously whilst it's very precise and reproducible, so you could pull samples together to make larger volumes, as Blaine was mentioning in our presentation, when you start looking at things like mouse studies and you need those larger volumes of production, that's where sunshine can come in to help transition to the next development stages of your particles. I don't know, Blaine, if you have anything to add to that.
Okay, I've got another one here. Bear with me as it's a little bit of a long one. I need to synthesize an LNP that I will use as a platform for mRNA delivery. Since I'm going to work with low scale, do you maybe have a solution that allows me to run some experiments of mRNA LNP synthesis with microfluidics without buying the big instrument for the larger scale?
So yeah, I mean the physically bigger of the two instruments, the Sunscreen, is the low volume screening instrument. So depending on what you specifically need, the smaller instruments can run a smaller number of samples, but your volume will be limited to a minimum of 1 milliliter. With the robotics and automation of how we aspirate our samples on the Sunscreen, that allows us to do some clever implementation of the sample handling that lets us reduce that volume even further. So if low volume scale is your priority, then the Sunscreen offers an ideal solution for that. If you're just wanting to run a few samples rather than many samples like you might on Sunscreen, then Sunshine can also run all of the exact same experiments, just with slightly larger volumes per experiment.
Fantastic, thank you. And I have an audience question here, which I think is for you, Blaine. It says, excellent presentation. Are you using PUC plasmid, and is antibiotic resistance a concern at this time?
So for us, yeah, I'm not going to comment too much on the specifics of our nanoplasmid construct, but no, that's not a concern for us at this time. Yeah, these are pretty well-validated constructs that we're using.
Fantastic, thank you. And next up, what microfluidic cartridge was used for the Sunscreen studies? And is the performance comparable for reverse angle and T mixer?
Yeah, is that question about this specific data set, or?
It doesn't specify, but let's assume yes.
Yeah, that sounds good. Maybe I can comment and then Ben can add as well. So for the experiments that I showed today, we're primarily using, there's a 100 micron junction chip that Unchained Labs has, and that we use that for both of our instruments, both the Sunscreen and the Sunshine. And that's, again, one of these nice things in terms of translating across the two instruments. We have found that when we move to other instruments with different types of mixing geometries, that you do need to do some troubleshooting in terms of determining like the total flow rate, the flow rate ratio that will work to get a particle that's within the same specifications when using the same lipid mix. So there is a bit of work that needs to go in if you change that particle formation step.
Fantastic, thank you. And Ben, did you want to weigh in on this one?
Yes, I think in our experience, because we have the range of different microfluidic sunnies with these systems, usually if you've got an existing process like a T-mixing process, you can find one of the sunnies that we have, which will give you really nice comparability. So that's one of the nice sort of flexible advantages there. If you're looking to have a kind of direct translation from an existing process into the kind of Sunny Suite family of products, then there's a very wide range of mixer options to choose from. And then once you have that kind of transfer in point, then you can look to see if any of the other mixer types maybe offer distinct or individual advantages.
Fantastic, thank you. I think we can squeeze one more question in, but as a reminder, do keep the questions coming because we can reply by email. So one more here. Does sunscreen allow for automatically changing lipid ratios or allow you to pick different lipids? And Ben, perhaps would you like to weigh in on that one?
Yeah, so the instrument is aspirating all of the sample from each of the two input wells. So yes, you can automate changing between different formulations and different ratios, but those ratios are to be individually prepared into separate wells on the plate. So what it's not going to do is pick up tiny volumes of lipid A, lipid B, lipid C, and mix those to create a pre-prepared formulation inside the system. But you can prepare those formulations using other methods and then add them onto the well plate so that when you start running your experiments, you can screen absolutely all sorts of different lipid formulations and cargo formulations simultaneously if you wish.
Fantastic, thank you. And Blaine, did you want to weigh in on this one at all?
Yeah, I can just maybe comment on, again, just saying that, while the lipid mixing step is something that we do ahead of time before we go into the microfluidic setup on the sunscreen, that still does save us a lot of time, and that we're able to kind of individually change the composition across the wells, set it up, and then walk away from the instrument while it does the formulation.
Fantastic, thank you. Well, thank you to Ben and to Blaine for answering those questions. I'm afraid that's all we've got time for today, but any questions we didn't get to, we will reply by email. The webinar will be available on demand tomorrow, so look out for an email from us with the link. And we'd also like to invite you to take a short survey to provide us with feedback on today's webinar. You can find the link to this either in the resources on the right-hand side of your screen, or it'll be in the on-demand email. So all that's left is to thank Blaine and Ben once more for a great presentation, and thank you to the audience for listening. We hope you'll join us again soon.